Hello everyone, welcome to our channel. Every day our cell face thousands of threats like toxin, radiation, routine metabolic insults, various infection. But normally our cell has a repair mechanism. They repair the various DNA damage through these repair mechanism. But what happens if these DNA repair system fail? So welcome to the world of genomic instability. Now let's discuss about the genomic instability. So by definition, genomic instability refers to uncontrolled changes in DNA structure or sequence from a single base to entire chromosome rearrangement. Uncontrolled changes in DNA structure or sequence. It can be vary from single base to entire chromosome rearrangement. Normal cell, after exposure to the various environmental insult, will result into the DNA damage. But with the help of P53, there will be a DNA repair. And whenever this damage is irreparable, then cell undergo senescence or cell death by apoptosis. Genomic instability is seen in the following condition: when there is mutation of P53 gene, cell unable to repair the DNA. P53 is the most common mutated gene in human cancer. Second is DNA mismatch repair defect. To understand this, we need to understand certain terms like microsatellite. What are microsatellites? They are 1 to 6 nucleotide tandem repeats. Normally their length is constant. Mismatch repair, whenever there is mismatch repair defect, which is known as microsatellite instability, there will be variation in the length, and this will result into the mutated alleles. Example of DNA mismatch repair is seen in HNPCC or Lynch syndrome. HNPCC stands for hereditary non-polyposis colon cancer, which is an autosomal dominant disorder. Mainly 4 genes affected: MLH1, MSH2, MSH6 and PMS2. Third category of or the class of genomic instability is nucleotide excision repair defect. When UV rays or are cell exposed to UV rays, there will be cross-linking of pyrimidine residue, and this will result into decreased DNA replication. In normal cell, this cross-linking is repaired by nucleotide excision repair. Whenever this mechanism is defective, there will be no repairing of this cross-linking, and this result into the development of certain diseases like xeroderma pigmentosum. Now the fourth category is homologous recombination repair defect. Covalent DNA crosslinks and double-stranded DNA breaks repaired by homologous recombination, and whenever this mechanism is defective, there will be result into the so many disorders, namely Bloom syndrome, which is an autosomal recessive disorder in which there is defect in helicase enzyme. Second is ataxia telangiectasia, another autosomal recessive disorder in which ATM gene defect in the ATM gene. Normally ATM gene promote P53 or increases the level of P53. Whenever defect of this gene or mutation of this gene, there will be no there will be no activation of the p53 gene occur. Fanconi anemia, autosomal recessive disorder in which repairs in double DNA cross links, hypersensitivity to chemotherapy in this disorder occur, and in ataxia telangiectasia hypersensitivity to radiation is there. Familial breast carcinoma are also an example of homologous recombination repair defect. BRCA1 and BRCA2 gene. The cancer associated with BRCA1 mutation is breast cancer, prostate cancer, and epithelial ovarian carcinoma. In BRCA2 gene mutation, this is also associated with breast carcinoma, ovarian carcinoma, prostatic carcinoma, pancreatic carcinoma, and stomach carcinoma. BRCA2 is also associated with male breast cancer. Another defective mechanism which result into the genomic instability is defect in DNA polymerase. Normally, whenever there is nucleotide mismatch, DNA polymerase pause and excise these mismatched bases and insert proper nucleotide. Whenever mutation of this DNA polymerase, this will result into the accumulation of numerous point mutation and increases expression of numerous neoantigen. Although these high neoantigen respond well to immunotherapy. Immunotherapy is a newer treatment modality in cancer treatment. Now we will see regulated genomic instability in lymphoid cells. Developing B and T lymphocyte express RAG1 and RAG2 gene products responsible for the assembly and functional antigen receptor genes. Whenever antigen exposed to mature B cell, they express activation induced cytosine deaminase AID, which is responsible for the immunoglobulin gene class switch recombination and somatic hypermutation. They associated with the AID induced DNA breaks and nucleotide substitutions and result into the translocation and mutations and development of the lymphoid neoplasm. So this is genomic instability for you guys. Thank you for your time.